Welcome back to Dawn Academy. Today we're going to be talking about Module 2.9 in IB Biology, which is all about photosynthesis. Now, this is an SL level, so that's good for you if you're SL. Great. Okay, so we're going to start off by talking about the chemical equation. So, photosynthesis is really the addition of carbon dioxide to water to produce C6H12O6 and oxygen. And there's also some side things which are very important, but they're not usually put into the chemical equation, like plus NADPH and ATP. So these are also produced, but they're not put in the equation, so don't put them in the equation, but just know that they are products. So we're going from carbon dioxide and water to produce glucose and oxygen. These are the main two products of photosynthesis. So basically, photosynthesis is a plant taking light from the sun and converting it into food, because plants are autotrophs, so they make their own food. So there's two types of photosynthesis. There is light dependent. So this is the one we were talking about, light dependent photosynthesis. And also there's another type called light independent. So light independent, as the name suggests, it's photosynthesis without the presence of light. So without the presence of light. However, there's also light dependent photosynthesis. So what is the difference between light dependent and light independent photosynthesis? Well, in light dependent photosynthesis, our end products are ATP, NADPH, and lastly we have oxygen. And in light independent, we use these products of light dependent, ATP and NADPH, to produce, in the end, we will produce glucose and we will produce oxygen. So let's go into the more general aspects of photosynthesis. So in photosynthesis, we have a plant, right? A plant is going to be doing the photosynthesis. All of it is by the plant. So the plant is taking light. Let's do this in highlighter. So the plant is taking light from the sun, right? So this is going to be the light source that is going to be able to facilitate this reaction. So over here, we're going to have light. Well, at least in the light dependent. So these rays from the light are going to come and shed some light on the leaf, and the leaf is going to absorb this light. The pigments are going to be really what is the actual biological aspect that is absorbing the light will be the pigments. So we have different kinds of pigments. We have chlorophyll, and we have A and B. We have carotenoids. I'm sorry if I spelled that wrong. Carotenoids, you can associate them with the color orange. And lastly, we have the xanthophil. Xanthophil. Might be with two L's. So these are the three pigments that are going to be found in plants. Absolutely, definitely. It's not like some plants have them, but they have them in different ratios. However, they are all present. So speaking of that, once we come to speaking about the electromagnetic spectrum, this is the wavelength. We have wavelength down here, and we have when we're talking about the absorption spectrum, we're talking about absorption. Percent absorption of light. Okay, so wavelength, we have to include it in nanometers, just to make our graph specific. All right, so we have different colors on the electromagnetic spectrum, and they range from infrared to ultraviolet. However, since we are taking light from the source of the sun, we're only going to be considering the visible light. which is composed of the 6 main colors: red, orange, yellow, green, blue, indigo, and violet, which you can do a mnemonic called Roig Biv if you want to remember it. So we're going to start our graph. This is going to be a very technical graph. We're going to do a break first in the graph because we are starting with 400. And we are increasing up to 700. In the middle here, we have somewhat of a 500, 400, 500, 400, 500, 600, 700. Yes, that makes sense. Make sure your graph is equally distributed. I think it's better if I put the 400 over here. Okay, and we have our percentage absorption of light. So we're going to start our graph. We cannot touch it like this. That's not going to work because it's not really starting from there. It's starting after this page break. So we're going to start our graph from here, and we're going to peak at around 400, and we're going to go down. We're going to have a trough at 500, and we're going to go back up and peak a little bit more at 700, and that's going to be it. That's our graph. So let's explain this graph a little bit. So why did we peak over here at around 400? Over here at this point? It's because 400 nanometers is associated with the color blue. And the color blue doesn't get reflected by the green leaf. So let's take a little zoom in on this green leaf. It's mostly composed of what? Green pigments, which is chlorophyll. So these are closely associated with each other. And when we have a lot of chlorophyll, when green light comes onto the leaf, it's going to reflect and it's going to bounce off. However, when we have blue light, it's going to be absorbed because our leaf isn't green. Our leaf is green, not blue. So the blue light will be absorbed. This explains the peak over here on the spectrum. Now, moving on to the trough. Over here, we trough at 500, which is associated with the color green. Since the green light is being reflected off of the leaf, it is not able to be absorbed, which causes the absorption spectrum to trough at 500. Now, going back up to this other peak over here, we're around 600, 750. 600, 750 nanometers are associated with the color red. Let me just label this as green. We're going to label this as around red. So that's around 600, 700 nanometers. And the color red will actually be accepted by the leaf a little bit lower percentage than blue, but that can be explained by the presence of carotenoids. Carotenoids are orange, and red is very close to orange. Therefore, it will be absorbed a little bit less than blue will. Okay? So that's it. That's pretty self-explanatory. Now, moving on to let's just put this, title this action spectrum. Feel free to take a screenshot and post it on Google. Okay. Now, moving on to oh, I'm so sorry. That is not the action spectrum. That is the absorption spectrum. Absorption spectrum. I'm deeply sorry. Delete that screenshot and take a screenshot again. All right, now moving on to the absorption spectrum. I'm sorry, the action spectrum. Now I'm confused. Action spectrum, but I'm sure of what I'm saying. Don't worry. Very credible source here. So we're going to have a very, very similar graph, and that is why I'm going to just copy paste the x-axis, because it is actually going to be exactly the same. Where is this? Duplicate. And we're going to have the same x-axis. So that's very easy for you to remember in that case. You just use the same x-axis, but the y-axis in this case is going to be the percentage rate of photosynthesis, which is similar to writing the rate of photosynthesis, but we're just going to write the percentage of the rate of photosynthesis. So same case here, we can't start from here because we have a page break. So we're going to start from here. And you're going to see that it's going to be very, very, very similar. And we're going to go down. We're going to go up a little bit more. And that's it. That is our graph. We can draw them like this part. It could be a little bit wider, but the examiners aren't going to be very strict about that. So we can widen this a little bit, maybe from here. Okay, that works, I think. And over here is going to be like this. Very, very similar. And we're going to explain that in a second. So also, repeating myself again, we're going to peak at blue. We're going to trough at green, and we're going to second a little bit of a smaller peak at red. For the same reasons, same leaf, but this rate of photosynthesis relies on the amount absorbed. That's why these two spectra are very similar. So if they tell you to justify why are these two graphs similar, you're going to say these exact words. The action spectrum is the sum of all absorption spectra. Now let's explain that a little bit. So going back to this action absorption spectrum, this is considering a green leaf, and we're considering chlorophyll. We are considering chlorophyll because it's the most prevalent pigment that is present in a green leaf. Therefore, when we say the action spectrum is the sum of all absorption spectra, we're talking about chlorophyll, we're talking about carotenoids, and we're talking about xanthophyll. That's why the graph looks slightly different down here, around this area, but it's not a very big difference that's significantly noticeable. That's why we say this phrase. It's a very important phrase, and if they needed you to justify your answer, this is what you need to include. And some other tips are, if we look at, let's say, a leaf changing colors, starts with a green leaf, and then it becomes red, and then it becomes, orange, then it becomes yellow, and then it falls off. This looks like a balloon, but I'm not intending a balloon. So it's changing colors. This is because as the temperature drops, also known as change in seasons, the temperature is dropping, chlorophyll starts breaking down. So it's breaking down, it's no longer chlorophyll. So when chlorophyll, the pigment, is decreasing, another pigment needs to be dominating. So in this scenario, from here to here, take a second, think about it. The chlorophyll is breaking, and this red pigment is taking over. Next, we go here. That pigment is breaking, and this pigment is taking over. It all depends on the optimal temperature of a leaf and how it functions best. Okay, next, moving on to our 3 factors. So we have 3 factors that affect the process of photosynthesis. They are CO2 absorption, or concentration is a better word to say. CO2 concentration. We have light intensity. And last, we have temperature. So I'm just going to draw these graphs really quickly because they're all pretty much similar. And I'm going to tell you something. These 3 graphs, these 3 factors are called limiting factors. A limiting factor is the factor that is provided in the smallest quantity that can slow down the process of photosynthesis. So if one of these isn't fully provided in the optimal amount, it is called a limiting factor. So over here, we're going to have CO2 concentration. These brackets mean concentration. You're going to have light intensity. Over here, we're going to have temperature. So CO2 concentration. Over here, we're going to have CO2 concentration. Here, we're going to have rate of photosynthesis. Photosynthesis. You guys know that means photosynthesis, right? If not, this is the rate of photosynthesis. And this is the same X for all of them. And over here, we're just changing it to light intensity and temperature. All right, now getting to the graphs, the good part now. So when we have not enough CO2 and we're increasing CO2 concentration, it's going to increase until a certain point over here where all of the enzymes responsible for CO2 acquisition, also known as Rubisco, are going to be occupied and working at their maximum capacity. As we learned in the topic of enzymes, they aren't going to be able to catalyze as many reactions, and therefore there will be a plateau because there will be a higher concentration of CO2 than the amount of enzymes. So the process will slow down and it will be less efficient, therefore it will have a plateau. Here it's in limiting factor because as it increases, the rate increases as well. Same exact graph applies for light intensity, for the same reasons exactly. However, in light intensity, it's not the Rubisco enzyme that is going to be fixing the carbons. It's going to be the pigments that are trying to absorb all the light. As the pigments are all satiated and satisfied, they won't be able to acquire any new light. Therefore, the graph will plateau. And here it's also a limiting factor. And in temperature, as we remember from our enzymes unit, we're going to have a slow increase. We're going to have a very steep decrease at the end. Over here, or we can draw it a little curvier, I believe. Something like that. So over here, it's a little bit slower. Here, there's an optimum temperature. And after that, over here, it's too cold, so the enzymes will be deactivated, as we learned in our enzymes unit. And over here, they will be denatured because the temperature is too high. And one last thing that I want to leave you with before I wrap up this video. The 3 techniques to measure photosynthesis. So we have 3 techniques. We have 1, change in biomass. Don't worry, they all get easier after change in biomass. Change in biomass basically means how much glucose there is in the plant. Second of all, we have CO2 uptake or consumption. This is basically how much CO2 is being uptaken by the plant to perform photosynthesis. If it's not taking that much CO2, it might mean that it's satisfied, or it might decrease the rate of photosynthesis. It's very interesting to see. And last, there is O2 production. As we reviewed at the beginning, there is O2 as a product of both light dependent and light independent. So it's a very useful thing to track when looking at photosynthesis. And that's all I have for today, guys. Thanks for watching.